Hi, everyone.
Welcome back. And I'd like to introduce the next speaker, Rebecca Morrow, who's going to talk about carbon cycling. Thanks, Rebecca. Best of luck.
Hi, everyone. I'm Rebecca Morrow. I'm presenting my thesis work on the effects of tidal inundation on carbon cycling in decommissioned salt pond soils. This work was conducted by the Blue Carbon Lab of Deakin University. So before I jump into salt ponds, I want to give a little bit of a background on salt marshes themselves. They're effective carbon sequestration systems, drawing down atmospheric carbon dioxide through autochthonous carbon input through primary production, and also through allochthonous carbon through sediment deposition and tidal influences. The highly water-saturated soils create anoxic conditions which slow microbial respiration, leading to low soil carbon loss through microbial respiration. So while there is soil carbon loss through carbon dioxide and methane, this is minimal compared to the carbon inputs through primary production in tidal deposits, leading to a growth in soil carbon stock, which can last for centuries to millennia. And this is effectively the biological carbon sequestration systems in salt marsh ecosystems. Now, these salt marshes are highly effective at carbon sequestration systems, sequestering more carbon than any terrestrial ecosystem. So you can see here it's on orders of magnitude higher than temperate tropical and boreal terrestrial forests. However, knowledge of how effective these ecosystems are is pretty new, and so these systems have experienced a massive loss of spatial extent, experiencing up to a 60% loss in the last 100 years alone, and up to an 80% loss in the past 300 years. Causes for these losses include sea level rise, commercial development, agriculture, and land uses like commercial salt production, as you can see here. These salt ponds are built on former salt marsh habitats, and they have low elevation and are close to the sea. In order to commercially harvest salt, they're continuously flooded with seawater. The seawater is allowed to evaporate, and then the residual salts are collected for commercial salt harvesting. These ponds typically have low soil carbon due to low primary production. As you can see from the characteristic red color, they do have hypersaline microbial mass, which is a common feature of hypersaline conditions. However, this primary production is incredibly low, and it's minimal when compared to natural native salt marsh vegetation. And there are tidal restrictions, obviously, which prevent carbon influences from allochthonous deposits. Due to technological advances, basically rendering these sites financially obsolete, these sites are pegged for salt marsh restoration to rehabilitate the ecosystem services that salt marshes provide. So these processes, these restoration processes typically begin with tidal reinstatement, reintroducing salinity conditions that are uncharacteristic of these salt ponds, or have been uncharacteristic for the past years that they've been in. And we've noticed in past salt marsh restoration programs that sometimes these sites can become temporary sources of greenhouse gases. Now, the data isn't there for salt ponds themselves, so we've just noticed that some salt marsh restoration projects can become sources of greenhouse gases. So there is very little data on what exactly happens in a restoration project in a salt pond system. So the aims of this study was to characterize changes to soil carbon cycling on the short term, and this will generate a preliminary picture of soil organic carbon cycling response to salt marsh rehabilitation programs. And so we're going to do that by characterizing changes in greenhouse gas flux over time. This will help us quantify the amount of soil organic carbon loss, and it operates on a time scale of minutes. So it gives us a pretty instantaneous picture of changes to soil carbon cycling. We're also going to describe the microbial organic matter decay rates using the T-bag index developed by Kuskamp et al. In 2011, which uses standardized T-bags buried at 5 centimeters below the soil surface, that we can compare the T-bag organic matter decay rates to different ecosystems across the world. And then we're going to characterize the dominant microbial metabolic pathways using redox potential as a proxy to understand how the organic carbon is utilized by the microbial community in the soil. So our study site was a decommissioned salt pond in Avalon Coastal Reserve outside of Geelong, Victoria in Australia on March 6th. So this is towards the end of Australian summer. We had two salinity conditions: our treatment course, which were incubated in 34 PSU water, which was the local seawater salinity, and our control course, which were incubated in 94 PSU water, which was the local pore water salinity. We had 8 greenhouse gas cores. 4 were incubated in our treatment, and the other 4 were incubated in our control PSU. And these gas fluxes were monitored continuously throughout a 30-day incubation period, some during the day and some at nighttime. We also had 30 destructive cores, 15 within our treatment and 15 within our control. These cores, we sampled 3 at a time, so we had 5 different sampling dates. And these were termed destructive because sampling of these cores required the destruction of each core. So at the surface, we took a 50 cubic centimeter sample for dry bulk density for carbon and nitrogen analysis. We also took pH and redox. At 5 centimeters, this was the depth at which we buried the tea bags, and at each sampling date, we collected the tea bags, rinsed them of sediment, and calculated the mass loss. And we also took a pH and redox measurement. And at 12 and 18 centimeters, we took our final pH and redox measurements in order to generate a depth profile in the soil. So we also did a bit of grain size analysis, and which revealed a dominance of silt and fine grained sediments. As we can see from the surface here, we had over 60% classified as silt and smaller according to the Whitworth scale. And throughout the entire depth of the soil, we saw a pretty consistent third of it being coarse or fine sand. So these are very fine-grained particles. Soils are typically associated with very low soil permeability and low porosity, which probably explains why, despite daily flushing for an hour of our treatment course with 34 PSU water, our pore water never fell below 66 PSU. So these are still really hypersaline conditions. And this is probably going to be a consistent feature of salt pond soils, as low soil permeability is desired for commercial salt production because it keeps the salt at the soil surface instead of being lost to deeper layers within the soil core to make it easier for commercial harvesting. So because of the persistence of the hypersaline conditions, we saw no significant difference between our control and treatment for the redox conditions, and we saw no changes throughout any of our 5 sampling days throughout the 30-day incubation. So this is an average of our control and treatment conditions for our redox depth profile. At the surface, we found redox ranges associated with aerobic respiration, which is to be expected. At 5 centimeters, we found redox and pH ranges associated with sulfate reduction, alongside these bands of soft black soil, which is characteristic of sulfate reduction. The surface and 5 centimeter depths did vary significantly from each other, so we could note that there was a change in the dominant microbial metabolic pathways. 12 and 18 centimeters did not differ significantly from each other, but these 2 depths did differ from the surface and 5 centimeter mark, and we found redox ranges associated with iron reduction, alongside these bands of reddish soil, which is typically characteristic of iron. These 2 processes are very important, especially when considering our gas flux. Here is our average methane flux. We found no significant difference between the control and the treatment, and in fact found a negative flux. Now, the sulfate and iron reductions are 2 metabolic processes that are more favorable than methanogenesis, which probably blocked the methanogenesis from occurring, preventing methane emissions in the soil. We did find a significant difference between our field measurements that we took on the day of the core collection and our control. We found a positive methane flux. As we remember from the satellite photo, these hypersaline soils do have a microbial mass, which is primarily typically composed of cyanobacteria, which becomes stimulated by light, generating a thin oxygen gradient, which stimulates the small methanogen community in the soil, which probably led to the differences between the field and the control treatments, which were incubated indoors under fluorescent lighting. And did include the values from natural salt marsh taken from the Bay of Fundy in Nova Scotia to just provide some context here. So while we did see a positive methane flux in the field, this was still much, much lower than a natural recovered salt marsh. We found similar results for carbon dioxide flux. Again, we found no significant difference between our control and treatment. However, we did find about 10 times higher carbon dioxide gas flux in the field. Again, this is light variation stimulating that microbial mat to which the aerobic heterotrophs had a chance to respond to. And here is just some context for natural salt marsh. These are some carbon dioxide emissions that we can expect to see. So we probably saw much lower gas emissions in our soils because the persistence of the hypersaline conditions increases osmotic stress on the microbial community, reducing gas flux outputs as they spend the majority of their energy trying to maintain osmotic balance rather than digesting organic carbon. Also, we found very low soil carbon. So this is the average soil carbon across all the cores. Again, we didn't find any pattern throughout any of the 5 sampling dates. So this is an average of between the treatment and the control, which we found no significant difference. But if we're looking at the natural salt marsh taken from the Bay of Fundy, we see that the soil carbon is much, much lower. So we've found no significant difference between our treatment and control between the 2 different teabag decay rates. So this is a collection of the average green tea decay between our control and treatment and rooibos tea decay between our control and treatment. So while we found no significant difference between the controls and treatment, we did find that green tea lost its mass at twice the rate of rooibos tea, as we can see from the K values here. It's because green tea is primarily composed of more fresh and labile organic matter, whereas rooibos tea is composed of more recalcitrant organic matter. While rooibos tea does have a hydrolyzable fraction, it's about half the size of green tea, which is coincidentally the size disparity in decay rates here. So that information combined with overall low decay rates compared to natural salt marshes, which can find K values of 0.05 or even 0.07. This is indicative of low soil carbon loss through microbial degradation and probably indicates that most of the mass loss we saw here was due to leaching rather than microbial degradation, indicating very low soil microbial activity. So what does this mean for salt marsh restoration scenarios? These sites will experience generally low soil carbon loss. And so this may mean, this is reflected in our greenhouse gas data and teabag data, this may mean that we may not see a spike in greenhouse gas loss or soil organic carbon loss, as microbial activity is generally very due to the extreme hypersaline environment. So this can be beneficial where in the carbon mitigation context. And this low soil permeability reduces the effective salinity, which may mean it will result in gradual changes in soil carbon cycling rather than abrupt changes in soil carbon cycling, as is common in other restoration scenarios. The persistence of hypersaline conditions may also delay plant recolonization, preventing more organic matter input through that highly valuable autochthonous carbon input that we saw from the first slide. Some limitations from the study: we greatly reduced or removed field conditions that we would experience in the fields within this mesocosm. So temperature is known to have an effect on microbial respiration. Increased temperature tends to accelerate microbial degradation and respiration. So we only experience this mesocosm only experience a temperature flux of about 2 to 4 degrees Celsius. So that was very minimal. As we also saw, light has a probably pretty big impact on microbial respiration. So because of these 2 effects, seasonality may put a large impact on restoration scenarios. Precipitation reduced or removed in this mesocosm. The amount of soil moisture will affect the depth of the oxic layer in the soil, which will either accelerate or decelerate microbial respiration, which again, we reduced or removed in this experiment, but we may see greater variation in the field. Additionally, we use artificial sea salt to make our seawater, which does not have its own microbiome. But however, tidal reintroduction will reintroduce a natural microbiome, which may affect how the soil microbial community recolonizes the soil after salinity conditions favor the recolonization. So a big thank you to the dedicated scientists at the Blue Carbon Lab, including Dr. Mel Wortman and Dr. Stacey Trebek-Tackett, and the rest of the staff. And thank you to IMBRSea for coordinating the opportunity.
Perfect timing. Thank you.